was certainly the sort of the bucolic life of a vet and mixed practice, meeting crazy farmers and crazy owners, and I definitely met some crazy farmers and crazy owners. They definitely exist. But the title is really sort of when I've gone from being a vet through to sort of a research career, and then I'll put it beyond because there's other things that I've done which are slightly beyond the research, but I think no less interesting. And I'm pretty passionate about encouraging more vets with clinical backgrounds into research. I think we offer a huge amount to the research community. So hopefully by the end of these slides, then I'll be basically going for, it shouldn't happen to a vet research, it should happen to a vet. So hopefully I'll persuade you over the series of the talk. I decided, going back to the beginning, is to have this talk a little bit in the 7 ages of man. And it was, so the first part is about conditioning. So if I wanted to be something other than a vet, it was going to be quite hard for me, because you'll see that I actually was quite conditioned at a very early age to be interested in animals and interested in research. I'll go into that in a bit more detail, thanks to my parents. Then sort of the vet training practice years into research and beyond, and I'm going to leave it to you to decide how far along I've gone in my life journey. Hopefully I'm not going to leave this lecture with a walking stick, but sometimes some days feel a little bit like I need one. So anyway, the other thing I'm going to do is go through the career, and I'll pop up the cards. Basically, it's a career of 7 mentors. I think this is something that's really, really important, is that I've had a huge guidance and advice and support from mentors. First one, obviously, is my dad's. He was apparently there from the beginning. And it's quite interesting to get the photos of, because I was trying to get photos of Gordon Harkis, who's my PhD supervisor, and Hugh Miller, who was head of school here on 2 occasions, as kind of key mentors before I moved to the Morden. I think it's because it's the era before smartphones, so I guess we have photos for everyone now. So I was searching for Gordon Harkis, and I was finding some funeral directors, but it wasn't Gordon. And then Hugh Miller, I couldn't find a picture of Hugh Miller at all, even though he has published like so many articles. Head of school twice, so if I'd had time today, I would have popped along to the dig deck, where there's a picture of all the different heads of school and taken a snapshot there, but obviously I didn't have time. But then moving on to John, who was my first boss at Morden, and Dave Galley, who I see, is Dave here? He's gone cycling, yeah. So David said to me, I'm in a real dilemma here, it's such a nice day, and I'm probably cycling a lot tonight, and I don't know whether I'm, should I stay for your seminar or not? And I said, well, it depends what you value most, So there you go, right? So if you just bear with me a minute, I'm going to take his picture of all my bloody snacks. Then on to Julie, who's obviously ex-director of Morden, and gave me a lot of sort of support in my career. And finally Sandy Trews, who I met through the vet record. He was also part of the board at Morden, but also, has been for a while, and now there's another vet in the House of Lords, but was for a long time the only vet in the House of Lords. He does a huge amount of work in terms of sort of trying to sort of change the political agenda and supporting animal health and welfare. So they're the career of 7 mentors, and this is the journey's beginning. So I was brought up in Stow, moved there when I was 2, when my dad moved up to Edinburgh. Both my parents are reproductive biologists, and we lived in this little village, and I got very interested in farming. And this was sort of on the left-hand side here. This is basically my journey to the local farm. So friends of ours had a farm here, mixed sheep and beef suckler enterprise. I don't think I called it an enterprise when I was a little kid there. It's a farm with animals and so I spent a lot of my holidays there after school, cycling along this road on a bike, no helmet. Of course, in those days your heads were much harder, so you didn't get too much help on the bike. And this was sort of the entrance into Luddick Farm. And I remember, so this is where I got really interested in farming, and I wanted to just be a farmer. That was my kind of like life's dream. But then I realised the farm's quite expensive, so maybe the vet that comes on the farm, that's quite an interesting job too. And I came back one day, and I was really excited because I knew some names of breeds. And one of the breeds I named was the classic Shirley Bill. So that's a Shirley Bill, yeah. I came back and Mom, yeah, they have Simmental bills and they have a Shirley Bill as well. So anyway, it wasn't as prime as that animal, has to be said. So then the beginning also not involved exposure to farming, but also involved exposure to research. This is my dad processing some samples in the lab. But basically, what we used to do is, because we used to go to the Marshall building, which is just at the old Roslyn site where Dad had his animals, and he worked on. It's very interesting. So the estrous cycle of ewes is quite similar to humans, and the anatomy of the reproductive tract is quite similar too. So it's a very good model to study, human reproduction. So that meant you got to see Dad chopping animals' heads in half to get the pituitaries, but also taking blood samples. And back in the day, you didn't have these disposable vacuum containers that you just threw into the bin after you'd finished them. You basically had glass vacuum containers, and one of the jobs I remember doing was they used to autoclave the glass vacuum containers, and then you basically put the stopper back in, and then my job was to get a vacuum pump and suck the vacuum out of the vacuum containers to allow them to sample even more. The other sort of interesting thing is I used to do work at the Marshall building sort of during the summer months, and it overlapped with the Dolly the sheep being there. So the animal techs, there was one particular animal tech, Nora, who used to take wool from the sheep and weave it into sort of wool to then knit scarves and whatever. And I remember one day we're in the sort of the coffee room, and basically she gives a handful of wool, and it's like, oh, this is Dolly's wool, so like, can you help tease it apart? And I wish I'd just said, can you knit me something like this one little scarf, so I'd have like Dolly's sheep scarf. I never did it, but anyway. Undergraduate years. This is me taken a couple of weeks ago. It's been a really hard, busy 2 weeks. This is actually the Glasgow University Prospectus in 1995, and it's thoughts from my 21-year-old self. So basically, in my vet degree, I took a year out after third year and did an integrated degree in biochemistry. Do not ask me any questions on biochemistry because it's gone in one ear and come out the other. But essentially, I did an integrated in biochemistry, and they were interested in sort of, in students who had done something slightly different with the course. And here are my thoughts here. It was great only to have taken 1 year out of my veterinary course to study for an honours in biochemistry, but I only recommend doing an integrated degree if you're very interested in the subject. And I think that's pretty true, but I think I say this to a lot of students, and and also just in life in general, you've got to really focus on stuff that interests you. If it's not interesting to you, it's pretty tough to do, to be motivated to do it, and you won't do a great job. So it's really amazing if you can do something in your life that you're really interested in. Had a bit of a dig at the clinical work here. So in the vet school, you didn't do much clinical work until your final year, which was no lecture free, but basically I was saying, can you spread it a bit more over the 5 years? And I think some of the courses are doing this now. But also, this was quite cringy. I didn't realise it was going to come into perspective. I also remember a rock band. So when I go ahead with my plan to work in practice for a couple of years, then take a PhD, I might become a rock star. And so anyway, I'm here. So basically, you can tell that it didn't actually happen for me. But anyway, the other thing was looking back on this is like, I was pretty laser focused on, I was going to go into practice. This is really my dad's suggestion, is like, get your clinical credentials and then go into research if you want to still do it. And so that's what I did, although I actually enjoyed practice a lot more than I thought, so it took me 4 years before I went to do the PhD. This is the end of the vet course, and this is me and my band. I did have a band. I would say that I'd learned the true value of having talent in music, and I didn't have it. But essentially, they had a sort of an appraisal, and my appraisal was a well-liked and appreciated member of the final year posse, which I think I looked at some of the other appraisals and they were pretty bad. So I think I got away quite lightly. So then from there, went into mixed practice, and I was a same practice, Willow Vet Group, which was in rural North Buckinghamshire. Mixed sheep, cattle, a few dairy herds, a lot of small animals. So it's about sort of 60 to 70% small, and the rest was large animal. Although it was only myself and the partner at the end that did the large animal work, so it was getting a bit more sort of 50-50. This is a contract of employment, and I was looking through it. They calculated your annual leave in hours, so I worked out that it was about 16 days a year that you had to leave. And that includes Christmases and New Year's and stuff. So I need to think about that when my job's quite stressful, but at least it's not 16 days. But essentially, I took it really seriously. This is my picture of when I first arrived at Willow Vet Group, and basically, I'm a bit of a prat, right? And my principal will basically agree with you on that one, but they had a series of cuddly toys as props, and I was like, just take a photo of me stroking this little cuddly toy. So that was on my parents' wall for a number of years, but no grey hairs in that photo, has to be said. So I was in practice, I was really enjoying it. You get a real thrill of actually, sort of you put all your clinical training into practice, and you're actually there on farms, on your own, making big decisions, doing things, getting client feedback, etc. After a while, you tend to find that common things are common, so you're seeing the same kind of presentations quite a number of times. And I was looking around, thinking that, could I do this for another 30 years? I was starting to think that I needed to specialize. It was fun, but I could see I was going to get bored. And the picture of it is a quick blood smear. So this is a blood smear you can just take in the back of the lab. Take a blood sample, you can quickly make a blood smear on a glass slide, put into the 3 stains, and then you can actually look at immune cells, which is where I sort of started to get interested in immunology. But I was looking for a specialism, and I was looking at clinical specialism. So the first one was like, maybe cardiology or dermatology or diagnostic imaging, but I couldn't really think of anything that I really liked, because I would quite like it more than anything else, because I like the generalism of practice. But I realised I was spending a lot of time in the lab that they had at the back of practice doing my own sort of diagnostics, and that's when I realised that actually my speciality is research. And I don't think many vets sort of think of research as a speciality, but I actually truly believe it is. I think it's as valid a speciality as being a cardiologist, large animal medicine, a small movement expert, because basically it's using your veterinary training, which is very broad. I guess vets know a little about a lot of things, kind of an overview of animal health, but actually you need the training and actually how to do research with the correct rigor. So I got a lot of advice from Dad about how to do this, and basically went around the different vet schools looking for projects. I'd had a friend who had a bad experience with a PhD supervisor, and so I was really keen on having a supervisor who actually would have enough time to sort of train me, and I wanted a project that I was really interested in. So Dad put me in touch with Gordon Harkis, and then started the PhD here at the Dick Vet, and it was all about respiratory transmission of this lentivirus, mydevisnavirus. At the time, it was not common. Now I find very few cases. I had to get my cases from Spain, which is pretty rife for mydevisna, doing my PhD, but now it is a real big problem. It's an iceberg disease, so you only detect, once you detect it, it's generally through your flock. But it's a chronic disease, it's incurable, it's really interesting biology. It affects macrophage cells of myeloid lineage. And when the macrophages are activated, you get bursts of virus replication, which is then controlled quite rapidly with an inflammatory response. So you get this kind of pulses of inflammation, and it causes this chronic inflammation. At the time of my PhD, there were 2 transmission routes. One was through the respiratory route, and the other one was maternal transmission, so from lambs ingesting infected colostrum or milk. And it was really, there was lots of kind of polarized ideas about which was the most important route. My PhD was really to address this research question about respiratory tract transmission. And the hypothesis that we had was that the trachea is uniquely sensitive to mydevisnavirus. Because there have been some studies looking at infection models showing that if you gave an intratracheal inoculation of the virus, you could get away with almost 10 infectious doses of the virus, virion particles, and it would cause rapid sort of seroconversion infection. That was my hypothesis, and I spent my first year bashing around with organ cultures from the trachea. I managed to get the organ cultures from the Marshall Building in sterile conditions, thanks to Dad again. And I could show that they could be infected with the virus, but it was really, really hard to infect them. And so then we sort of thought in my second year, let's just have a look and see, well, what happens to this inoculum that we're giving to these sheep? And what you can see here is, here's a picture of a sheep lung here. This is where the inoculation site is. You can see that actually intratracheal inoculation is just a really good way of delivering it to the lower lung. And you can sort of see that it sort of spreads down into the lower lung of sheep. So then that got us thinking, we could actually model this. We could actually test to prove what is the main route of uptake of the virus in the lung. And so this is where I sort of collaborated with Dave Colley. Here, he was basically based at the Dick Vet. He was using sheep lung as a model of delivery of cystic fibrosis gene therapy. So formulating the particle size, the type of DNA plasmid that would be most efficient for uptake, and that was used to parameterize dose rates for humans. But essentially what we had here is a model here. We could install the dye into the trachea, so expose the trachea, and because you've got this mucociliary escalator, basically your inoculum would just move up to the larynx and then be coughed and swallowed, and you didn't get any exposure into the lung. And what we can nicely show here is that if you give intratracheal inoculation or a lung installation, you get infection. This is weeks to see PCR positive in the white blood cells in the circulation, or weeks to see your conversion. IT and lung installation are pretty much the same, whereas the tracheal installation is really inefficient. So then we proved that actually it probably is that what we're doing here is that virus is getting in, it's getting into the lower lung. We then showed that there's a lot of free virus in lung fluids, which was not known at the time as well. Then we could do experiments where we really push the sort of envelope here. We could take macrophages from the lung, infect them ex vivo, put them back into animals and track them in vivo, and show that that's a really good infection route as well. And then we could also do an early foray into transcriptomics using the kind of quite nice models that David had developed. So basically you have different lung lobes, and you can sort of do pre-challenge lavage to get the alveolar macrophages. You can then sort of challenge challenge lung lobe, a control lung lobe, and then come and lavage them afterwards. And then look for kind of changes in cell types, gene expression. And this is my early foray into transcriptomics. At the time, RNA-seq wasn't a thing. We were trying to do microarrays, which were really painful. But I did develop some cytokine quantitative PCRs, which was the cutting edge. And I was really proud of this result to show that actually the virus caused this upregulation of GM-CSF in the alveolar macrophages. That meant that we came to this kind of final model. It's quite a nice conclusion to the PhD. So what we thought was free virus was getting into the lung. It has been taken up by alveolar macrophages or directly to dendritic cells. This would uptake of infection would cause increase in GM-CSF, which would activate the macrophages and also local dendritic cells, which would cause increased virus replication, and that's how it's getting into the body. So that was my PhD, and I never worked on MIDI again because I moved to Moreton. But then a few years later, this was during the pandemic, a couple of guys, Nick Saville had done some modelling work on ESTA E. coli 157 a few years ago from King's Buildings, and Andrew Ilias, who was an old evolutionary biologist who had a flock of sheep that had infections were infected with mycobacterium. Andrew Ilias didn't really believe what his vet was telling her about transmission routes. And then with the data that was generated in my PhD, he basically started doing some modelling studies with Carrie-Anne from Royal Hede in the Netherlands. And basically what they sort of showed was that transmission is 1,000 times more likely in house versus grazing sheep. So basically it's all about contact. And they also showed that this transmission through milk is just almost negligible. So that was kind of like just a nice circle back to say that actually, the results I did in my PhD actually sort of were really useful a few years later and sort of proved this kind of that it was respiratory transmission driving a lot of infections. And this was the height of COVID, and we got a lot of press because we were talking about social distancing sheep. We didn't ask people to put face masks on their sheep, but so what did I learn from this? I think I learned that hypotheses are there to be tested, and because I spent a whole year trying to, basically my whole hypothesis of my PhD was proven wrong. Hypotheses are to be tested, and you just want to get the right answer. Also, to lean into my clinical skills, so a lot of the work I did was more clinical, but also to be collaborative and not be afraid of new technologies. Because at that time, you probably don't believe this, but at that time, I did sort of 12 cytokine quantitative PCRs, and people were saying, "Oh, that's far too complicated, we'll never get that to work." And of course, nowadays you just do send off to Node, you can get it 2 weeks later, and you've got all the cytokines, haven't you? But anyway, so yeah, that's in the dinosaur age. Then Morden, I came to Morden, and this is the beginning of the Morden years. 2005, straight after my PhD, I gave myself a nice 5 days off and started at Morden. And this was really because Hugh Miller, who is faceless, apparently he's someone who's, maybe he's on the dark web, I don't know. Anyway, so Hugh had suggested, because I was asking him, where should I go for a postdoc? He said, I think the Morden's a great place to go because it's very translational, but also there's a really good guy there called John Huntley, who Hugh had worked with on mucosal immunology. And also this guy called David Galley is really into cycling, so he doesn't like staying around for his mate's kind of seminars, you know. Anyway, joking aside, so David was sort of the rising star of E. coli 157 research. Molecular bacteriologist, extremely brainy sort of guy, I think you'd agree. But essentially, the postdoc was to develop these E. coli 157 vaccines. So E. coli 157 is basically it's an important zoonotic disease. Because of the toxins it produces, they damage the blood vessel linings of humans, and it can cause pretty severe diarrheas, but it can also cause irreversible kidney failure and also neurological signs. And it's really a motive that can cause death or high morbidity in young kids and also in older immune-compromised people. So it's not, the incidence rate is not high, but the impact is massive. Cattle are very interesting, and ruminants in general. They can be colonized with this bacteria, but they're insensitive to the toxin, and so they don't show any clinical signs at all. So the idea here is if you vaccinate the cattle, you can reduce the amount of bacteria that's produced in cattle feces, and that will sort of reduce the risk to humans. And so that's what we did. We developed a recombinant subunit vaccine for 157, and it was based on some really precise knowledge about the molecular mechanisms by which the bacteria attaches to and colonizes the gut. So it's got these flagella here. These are sort of like it allows it to swim around, but also the flagella then sort of dock to the surface of the epithelium. And then once they dock, then they switch off the flagella expression. They turn on the type 3 secretion system, and that is involved in this initial intimate association with the epithelium, and then they form microcolonies. So we came up with an approach which we called sequential antigen targeting, which is basically targeting more than one bacterial colonization mechanism. So here's a sort of schematic here. I had to pick this cartoon because it took me bloody ages to make it, right? So here we are, the flagellin, the bacteria is coming in, it docks to the epithelium, it stops expressing flagellin, it starts expressing the type 3 secretion system. And then this is the really neat bit. It uses this kind of, forms this needle-like structure, which then pumps effector proteins into the host cell. And this is what I'm indicating here in the red. So, and one of the effector cells it secretes into the host cell is the translocated intimin receptor, or TIR. That goes to the surface of the host cell and binds to intimin on the surface of the bacteria, and that's what causes the intimate association. If you knock out the type 3 secretion system, you don't get colonization. So what we did is we targeted H7 flagellin, intimin, TIR, and SB, and various combinations of that, and developed a very highly effective vaccine. So this is just the vaccine here. This is looking at animals that we've vaccinated and challenged with a dose of 057. This is a control group here, and this is our vaccinated animals. You can see it completely reduces the total bacterial shedding, and it also stops a large proportion of the animals becoming colonized in the first place, so it really blocks that initial interaction. So then we managed to get some funding to evaluate the vaccine in GMP conditions in a USDA feedlot with Jim Bonner here, and that was through Roslin Technologies funding. To cut a long story short, it's one of those results. It kind of sort of worked, but it didn't work well enough. So it was confounded a bit because we had different prevalence rates at the start of the vaccination program. So the controls are in the blue and the red are the vaccinated. We did see some effect, but it was temporary. So at the moment we're kind of like building up the energy to repeat this trial. It requires probably different adjuvants for increased duration of effect, but also balancing the groups better to start off with. It was one slightly out of our control. This was done in lockdown. Further adventures in STEC, I had to put a picture of Stephen there, a young Stephen, I would say. So one of the interesting things about STEC is that in Scotland we have much higher incident rates of human infections, and we think this is because we have a particular strain that dominates this phage type 21-28 strain, which often contain a toxin type, STIX2A, which is more virulent than the other toxin types. And this is a huge amount of work funded through FSA and FSS, but basically what we could show is that these phage type 21-28 strains were pretty transmissible. And if you take a strain that doesn't really is not very commonly infected, seen in cattle or in humans, that they don't really transmit at all. So this is, if you look at direct challenge, this is the phage type 32 strain. It colonizes less in animals. The phage type 21-28 strains, either with this 2A toxin, which is a really virulent one, or with or without the 2A toxin, these are isogenic. But they colonize about the same in direct challenge. But look at the difference in transmission. If you have a 2A toxin, it basically transmits really effectively to its pen mates. The phage type 32 doesn't do anything, and the 21-28 without the 2A doesn't really work very well. So what we could then sort of show is that this is being selected for probably because it's just very good at transmitting between animals. And then a foray into enteroids, the organoids, which Neil Navas sort of generated here at Morden. But essentially also what the phage type 21-28 strains did was with their toxins, they affected the stem cell turnover. So that gave us this kind of model here that the toxins are very good at colonizing animals because what they do is they inactivate the stem cells, and that slows down this turnover of epithelial cells. So if you think you're a bacteria here colonizing, if the cells turn over, then you're basically going to sort of like be shed off and then sort of excreted. Whereas if you can slow down this turnover rate, then you can get sort of more persistent colonization. So that's a really nice story explaining why we seem to be selected for STIX2A. It's very transmissible, and it colonizes very effectively. And also, worryingly, it's the most virulent toxin type. So, further adventures, that led on to some interesting projects. We looked at another FSS-funded project looking at the risk of Shiga toxin-producing E. coli, STEC, if you're cool, you can call them STEC, in wild venison. And this is because there was an outbreak of STEC associated with consumption of wild venison products. I think it was like burgers and sausages. And FSS wanted to understand the risk of wild venison to humans. So what we did is we actually empowered deer stalkers across the whole of Scotland. We sent out sampling packs. They sampled the deer when they were growled on the hill. That's where you kind of shoot them, sorry kids, to control the population, and then you can sort of basically they take the contents out of the intestine. They took a sample there. That's where you get your wild venison from, and that's most of the venison that we consume in the UK. But it was a really amazing sampling effort. So we had Forestry Land in Scotland sample, we had samples from deer management groups and individuals, and we got a sort of spread of samples all across Scotland from all the range of different species. We only found 3 positives. What's quite interesting is that out of the 3 positives, they're all this 2A subtype. And also, we hit one deer isolate from the same location that was a human outbreak. And the human outbreak was associated with people walking in that area, sort of walking or biking in that sort of area of Scotland. And it was this postcode here pretty much tells you it was exactly the same strain. So the conclusion from this was that, well, I guess 2 things. One is the power of genome sequencing was pretty amazing to get this needle in a haystack. But the conclusion was that wild venison is probably not high risk, but it potentially can carry something quite dangerous. So what did I learn from that? I think, again, it was more collaboration was really, really important. I think the other learning, particularly from the wild venison project, is that if you want to do policy relevant research, it's really easy to do it if you're doing it with a co-constructed project from an agency or a policy team. Because basically, instead of you trying to sort of persuade somebody that your research is really important, they're basically sort of like trying to bite your hand off to get the results. And that was kind of really, really important. If you're at the Morden and you know anything about the Morden, you realise that parasitology is quite a big thing. So being at the Morden, being involved in vaccine development, and being surrounded by parasitologists, you couldn't really escape, so I got dragged into the parasitology field, which is actually really exciting as well. So we work on parasites that affect the stomach of sheep and cattle. We study parasites that affect the small intestine of cattle. I was trying to work out how I actually sort of got into parasitology. I think it was almost by, not osmosis, but I slowly, bit by bit, became more of a parasitologist. And it was partly because I think John was just really good at supporting me and giving me this sort of space to kind of explore different sort of areas of research. So the first sort of area I was quite interested in was because I was interested in mucosal immunology. I'd done a lot of work looking at E. coli attaching to the epithelial surface. It was about this kind of the way that parasites can change the immune system, modulate it, subvert it, so they can survive within the host. Because if you think about it, this is a parasite, a telangiectasia parasite in an infected gastric gland, and it's got a lot of an immune response all around it that somehow is managing to survive. And it does this through secreting lots of different factors which really mess up with the immune system. And so what we could show is that, for example, we could induce these parasite infections were really good at inducing a T cell response, a regulated T cell response, so more sort of T cells that down-regulate immune responses. And also they can switch production of cytokines from a sort of more inflammatory to a more regulated response. I'll put this slide in here because this is from an honors project that I did, and I don't know whether you remember, Mum. I took a slide to you and you did it on the confocal at PMRI. You don't remember that? I guarantee you definitely did. Because I took that, I could take an image like that. And then this is the old Macaral, who I've worked with closely for years. Like, I know we look really young, but we've been working together for a long time. But then Al was very interested in developing vaccines for roundworms, and this is the telangiectasia circumcincta vaccine. Essentially, what the vaccine is a suite of antigens that are either immunostimulatory, so they're targets of the immune response, the IgA response, which we knew was correlated with protection. So Al had sort of identified and cloned and expressed all these candidates. And then also there's interest in immunomodulatory vaccine targets, because if you could try and neutralize the function of an immunomodulator, you can kind of take the brakes off the immune response. So there's a combination of stimulating the immune response and taking the brakes off the regulation. I remember Al and I were in the ethical review committee and trying to get the first experiment through, and the committee was saying, well, this isn't really going to work, is it? It's never going to work. And so we still had to make the case that, well, we don't know until we tested it. And of course, the bloody thing did bloody work. Yeah, so basically, this is the fecal egg counts from infected animals, and you can see the controls here and the vaccinated in here. So clearly seeing quite a big divergence in egg counts. And that was the first example of a roundworm vaccine that was induced protection with a recombinant protein. So it's a bit of a milestone in vaccine development for roundworms. So moving on to sort of like more, you can see I'm kind of taking each kind of like research area and moving through through the gears here. But I had a long time with my tracheal organoids, like thinking that basically they're just dying, and I was never very happy with that model. We played about with sort of enteroids as well with the S-Tech, and I thought they were really powerful technologies. But I was always looking back to the O157 vaccines, thinking if we could only have a way of precisely understanding the molecules that are involved in parasite establishment and survival in the gut, then would basically be a much better way of rationally selecting vaccine targets. And so this is where David and Mark, both in the audience here, developing organoids, and based on the work that Neil had started here at Roslin. So essentially the idea here is you can take stem cells from animals, and then in the right media conditions and the right matrix, they will form three-dimensional organoids, which kind of represent the cell types and part of the orientation of the tissue of origin, and then you can use those to study infection in a sort of precise way. I think I need to stop the pointer, because I've got to show, Mark likes his videos, right, so I have to show a video here. So this is basically just an isolated crypt, and it's showing that once you've isolated it over a few hours, it just forms this nice sort of like structures, and then it will expand, and you can passage it, you can freeze it, you can resuscitate it. And it means that not only can we use these systems to understand kind of precise host-pathogen interactions, but we can also use less animals in research, which I think is something we all need to sort of strive for. So the other really great thing about these organoids is when we took our parasites, these are the infectious stage of the parasites, what these parasites do is they seek out and invade the organoids, just like a parasite invading a gastric gland in the actual animal. So we think they're a really good and valuable model system to look at these precise interactions, which is what we're currently doing now. We're seeing lots of interesting biology, and we're trying to understand the molecules that the parasites are producing to induce that biology. So this is just some other sort of, to round this home, but this is a work project with Mark, Al, and then Neil and Liam, who are also in the audience. So we managed to get funding from Zoetis and basically develop these organoids to look at Ostratasia, which is a roundworm of cattle. And this is some nice pictures that Mark produced showing that this is the tissue, and that's an Ostratasia L4 right next to the epithelium in the gastric gland. And this is our organoid model of that. And you can see here we've got sort of mucus layers in our organoids, and this is in vivo. So we think it's quite a good model system to study these interactions. The other thing that's increasingly becoming complex is the space matters. So I thought RNA-seq was quite interesting because you could take a bulk as a tissue sample and you can get some genes that are upregulated or downregulated to understand the biology. But actually it's the spatial context that is really important as well. So here we have an infected gastric gland, and you can see there's the worm in there. The gland is very expanded, and we see that phenotype in the lab. But also we can see the epithelium is de-differentiated, so the cell types we would expect are no longer there, so the parasite is stopping the epithelium from functioning properly. And also you're getting like a lot of immune cell recruitment, so the conundrum has always been in these parasite infections, is you get a really rapid immune response, but actually it is not protective. And so I think what we're thinking here is that the immune cells can recruit to the site of infection, but they can't get close to the parasite. So if we can subvert that interaction, then we can hopefully sort of generate some more effective control measures. This one is now about moving to population biology, and this is through guidance of Julie Fitzpatrick. So after my postdoc on 0157, I was like, well, I'm kind of looking forward to looking to move to do some probably more than I'm going to move somewhere else and learn some other skills. And I was looking at fellowships, opportunities to move maybe back to the university. But actually Julie sort of said, well, actually there's a potential space available for you at Morden, a role, but it's in population biology. I'm a member of the human population, but that's about as much as I know about population biology. So at that time anyway, but it led to some really interesting projects looking at sort of population-based immune phenotyping. And the first one we did was looking at immune phenotyping of the Langham dairy herd, and this is because it had some very defined genetics. It had control lines, which were sort of the average genetic merit of dairy cattle across the UK, but it also had selected lines that were selected for increased milk yield and quality. And what we then did is to develop these panels of immune phenotypes in serum, but also in the cells, looking at the population structure of the circulating immune cells. And you can see some really interesting interactions. If you took those, well, first of all, you can see differences in the lines. And we went on to show that a lot of these traits are heritable, so under genetic control. But also, we could identify some of these immune traits correlated well with disease, and then could potentially be used for selection purposes. For example, CD4-CD8 ratio was associated with clinical mastitis. So that was a really interesting foray into population biology, and I've continued to do sort of work in that sort of area, looking at for markers of disease resistance. And this is Dan Nussey, but this led directly onto projects on Syncytial soy sheep project, which once you start working on the soy Syncytial project, you never seem to be able to escape it, regardless of how hard you try. But anyway, it's down to this guy here, Dan Nussey. He came to Morden because he was interested in parasites and wanted to know a bit about parasite immunity. So I had some chats with him and decided about some projects we could come up with. And one of the projects that we did was based on the fact that each year they catch the animals in nets. It's quite interesting. You have to basically collect the sheep. You put out nets. That takes you about 2 days. and then you spend you act like sheepdogs, basically running around trying to gather all the sheep into the net, and then you can do things like take weights, look at sort of take a blood sample, and you use those blood samples to then do some immune phenotyping. We showed some quite interesting things. So Dan Nussey was very interested in senescence. And what we could show in these populations, you get changes in immune cell population over time. So you're looking at sort of signatures of immune senescence. And cut about 10 papers out of that. We focused on parasite-specific antibodies because they seem to be predicting mortality in the sheep. And this is basically, what this is showing here is this is a parasite-specific IgG, and this is years before death. Sorry kids, the animals did die. They didn't go to the little farm up the road. But essentially what you can see here is that as an animal approaches death, then basically its antibodies drop to the parasites. And it wasn't just the mean level of antibodies, it was the change in antibodies. So if an animal changes from its mean level and then it goes down, it's the rate of change that predicts death. So actually this is probably the most convincing evidence of immune senescence in wild animals. And actually it's very hard to do. And the reason we could do it is we took data from 1995 to 2015 and just did antibody measurements on all that population. So we've got like lifespans of individual animals. This is probably the only time I'll ever have a science paper, but it was like, one of those that is really amazing result that came out of it. So it just goes to show that sort of, population-based assessments are really useful. Yolanda is postdoc has been with me for a long time here. We've taken this bit further forward, so we've done more detailed immune phenotyping, look at T helper cell types, the cytokines they produce, and we can see really neat things proving that Th2 cytokines like IL-4 are associated with worm resistance. So I'm going to sort of stop there, but really sort of useful and really exciting and interesting work. So I just thought I'd finish off the research sort of bit to look at the future of livestock disease research, and I think it's just a really exciting time. In the past, I would say that if you didn't work in a model species, like if you didn't work in mice, you wouldn't have the reagents to be able to do any of this kind of very precise sort of host-pathogen interaction research. But now we can do single-cell seq on cells. This is from Kate Hildersley's PhD. We can do in vitro models. We can generate, and we're at a similar sub-level to the human and mouse field. We can look at spatial transcriptomics. So we've got the tools now to look at the transcriptomes at particular sites in the tissues. But also importantly, we can also test it in a relevant target species. So I really believe that that's where all the excitement and innovation is. I think the days of just focusing on mouse models are kind of hopefully more numbered. So I guess what did I learn from this experience? Again, it was like I think it was really about collaboration. It was about interesting and being curious. And I think also just there's so much potential in research in animals. So that was the main sort of learning objectives there. So beyond research is just the final section of my talk, and the first bit I have to mention. I think I mentioned at the very start that I'm really keen to promote clinical vets going into research. But this is one of those. I guess it really helped me in the long run, but at the time it felt a bit like a hospital pass, because I was asked to chair out the blue a session at AVTRW, which is the Association of Veterinary Teachers Teaching and Research Work, their annual conference, by David Ecclesall, who coincidentally taught me biochemistry at vet school. And I thought, okay, I'll chair the session. But essentially what they did was they got me really drunk at the conference dinner and then said, you know what would be really good for you is if you became the junior vice president of AVTRW. So I was like, all right, okay. So obviously after a few glasses of wine, I was like, yeah, excellent, whatever. And then my first day in the presidential role, the treasurer said to me, oh, by the way, we're going to go bust if we carry on going to having conferences like this. So we need to come up with a different strategy. So it was an interesting time, and it basically meant that we had to restructure the whole organization. we now go around the different vet schools. It's much more focused on early career researchers, but it was an interesting time. I made a massive nuisance of myself because I was emailing the vet schools councils, getting all the heads of school to support the AVTRW. Stuart Reid at Royal Veterinary College was really supportive. We put our first meeting on at the Royal Veterinary College, but it basically made me known within the sort of the veterinary sort of profession. And that led on to this part of my life, which is the vet record and veterinary politics. I love the vet record, I'm not sure about the veterinary politics. But Suzanne Jarvis came up to me at one AVTRW meeting and sort of, I think said, well, we've got an opening for the clinical research editor of the vet record. Would you be interested in that as well? And I think I said, I have no time to do it. But then, of course, I had a chat with Dad. He thought it was quite a good thing. It would be a good opportunity. So I took the role on. Again, it took a bit of restructuring because they expected one person to do all the associate editor work. That would be sort of a normal panel of like 7 to 10 associate editors. There were no associate editors. You had to do all the sort of handling of manuscripts themselves. So I said, look, I can't do that. So basically, we restructured, and that structure is still in place at the vet record. But this is where I also got to know Sandy Trees, Lord of the Trees. But Sandy's been really supportive as well, always there for advice. And also, you know, sort of, I think as vets, we need to be a bit more visible in politics and shaping policy. As I said, he was the only member of the House of Lords that was a vet. And I think that we sort of see that again and again, that representation of our profession is, in political circles is not really high. But that was sort of, you know, it was originally painful, but I think it really opened me up to being involved in a wider sort of veterinary community. So I guess what did I learn from this? It was to be open to other opportunities. It was to be aware that, you know, sort of it's not just focusing on research, but actually trying to influence the veterinary profession in a more general way. And also the importance of restructuring. I think structures really sort of help organisations work. And it sounds quite boring, but if you get the right structure, then generally things kind of tend to work quite well. This is my management beginning in 2016, as Lisa said. With Julie's kind of advice, I took over the head of pillar at, as it was known at the time, for disease control at Morden. They were called pillars, and then people kept on calling us head of pillars, you know. Or then the first iteration was like main advisor, or do you remember this, Mars or Das? Main advisor or deputy advisor, that was the first iteration. So I think Philip Goose said, are you my ma or my da? You know? Head of pillar, became head of department. This was actually the first head of department meeting here. I was trying to make a play on disease control. It's a very bad pun joke, I'm sorry about that. And then to the change at the top, became the director of Morden in 2023. I think it was really a good experience in that, you know, sort of Al and I were taking a more senior role when Julie became Chief Scientific Advisor for Scotland. So we were onto the Morden boards, and we learned about how the operation of the Morden group works. I like this picture. This is the last picture of Julie before she retired. It's her last speech. And I almost look like I'm just getting ready to sort of like push her out of the way. But I had a better plan than that, because basically what I did is I got a really nasty alpaca and attacked her at the Royal Highland Show. So anyway, so that's the change at the top, and that's where I am now. Just to sort of say a little bit about what I think about the global challenges for the livestock sector. I think probably most of us know this, but one of the big issues is that, you know, as a sector, particularly in the ruminant sector, is the impact of livestock farming on the environment, which is really, really important. So methane mitigation, making animals more productive to reduce methane emission intensity, but also sort of, you know, really sort of supporting the industry as they kind of move to a sort of like a more environmentally sustainable farming system. The other sort of big concern for us at the moment is the climate change is happening now. and that's changing our livestock disease distributions. So for example, these are midge-borne incursions, disease incursions into the UK, so Bluetongue virus 8, Schmallenberg, Bluetongue virus 3, and now Bluetongue virus 12. And this is the Haemonchus contortus. This was taught to us as a tropical parasite of sheep and goats mainly. And now it's basically spread all the way up to the north of Scotland. And I think Rob was saying today that there's cases in Harris. So these are sort of challenges for us. You can see that over the 7 ages of man, I've come to this sort of left slightly, it's way too long towards the end. I haven't quite got to the walking stick. And then this was just a sort of final picture for me, is that it's a family affair repeating itself. I just saw this picture in the house, and this is, I think Dad and I are the same age. I think we're 50 or something like that then. You see the hairstyle is basically the same, isn't it? Some things don't change. But essentially, this is Dad showing Princess Royal around his lab, and this is me showing Princess Royal around the moors. And I think I've got far more grey hair, so I don't know what's going on. Anyway, so this was in the height of November. Apparently this is a really privilege to get a photo like this, just the two of you looking at the camera. But God knows what I'm doing with my face. I look like I'm sort of like Wallace from Wallace and Gromit. So that's the end of my life. Thank you for listening. Lessons learned. I think I just saw a few little lessons here. I did have on all those what I learned slides. I had notes to kind of like, so I nailed all the lessons learned, but I freestyled it all the way through because I don't speak a presentation together. Anyway, that's a technical issue. But these are the main lessons I learned. The first one is be curious. You've got to be curious. You know, we are really privileged to be able to work in an area that we are actually allowed to be curiosity driven, and we can do stuff that really excites us. Be positive. I think for me that's really important. I think it's easy to be negative, but I always try and see the positives, even though, as Catherine would say, I usually go through a trough of negative before I see the positive. Generally try to be positive. This is a really important one. Be kind. I think that's really important in life in general. In collaborative research, just be kind with one another. I think I could paraphrase this and say, don't be an arsehole. That's what I say to my kids quite a lot. Don't be an arsehole. I say, well, you're being the arsehole. Be collaborative and embrace the expertise of others. So basically, I guess what I'm saying here is that the successful research is through collaboration. But it's also like, you know, people have amazing skills and expertise, and I always feel a bit out of my depth a lot of the time when you're speaking to people who've got such amazing skills and knowledge. But it's basically just embrace that, you know, and collaborate. Don't be afraid of asking silly questions, and I think probably there are no silly questions. They're just questions. I've asked a lot of silly questions over my life. And then I think just to bang on about the clinical vets, I think we do need more clinical vets in research. So, from practice to research, I think it should happen to vets, and a lot more of them. And I'd just like to say thank you to everyone who's been with me on the journey, Catherine, Will and Tilly. Family and friends, colleagues and collaborators, mentors, funders, University of Edinburgh, give me this honorary position, and thank you for all for listening.